Hello everyone, I'm Rajeev Mishra. Today I'm going to talk about my master research project, effect of maternal immunization against myostatin on the post-hatch growth performance of their chicks. Myostatin is the member of transforming growth factor beta superfamily, and it negatively regulates the skeletal muscle mass. Studies have shown that suppression of myostatin significantly increases the skeletal muscle mass. A study from our lab shows that when monoclonal anti-myostatin antibody was injected inside egg yolk, it increases the body weight by 4.2% and carcass weight by 5.5% as shown in this table. So we hypothesize that when a hen is immunized against recombinant myostatin, it's going to produce anti-myostatin antibody, which will suppress the myostatin activity and increase the muscle mass of the broiler chicks. For this hypothesis, we have three small experiments. I'm going to talk about the experiment and the results side by side. Antigen preparation. For this, we have two antigen, recombinant myostatin and myo-2 peptide. For recombinant myostatin, chicken myostatin cDNA was introduced into PET45 plasmid, and this plasmid was inserted into E. coli. STS page analysis shows that when transformed E. coli was induced to using IPTG, it produces the recombinant myostatin as an inclusion body protein. And the inclusion body protein was further processed to produce recombinant myostatin, and the soluble fragment of recombinant myostatin was used as coating antigen for ELISA. The second antigen is myo-2 peptide. Myo-2 is a 15 amino acid fragment of the whole chicken myostatin sequence conjugated with KLH. And this portion of the myostatin binds to the activin type 2B receptor and initiates the myostatin signaling process. The second part is hand immunization and antibody titer evaluation. For this experiment, we have three groups of birds. Each group have 3 Highland brown female and 1 white leghorn male. The control birds were immunized with phosphated buffer-saline with adjuvant. The recombinant myostatin group was immunized with 0.5 mg of recombinant myostatin with adjuvant, and the myo-2 group was immunized with 2 mg of myo-2 with adjuvant. The blood sample were collected on weekly interval for antibody titer determination. ELISA performed with blood serum shows that peak anti-myostatin antibody titer around day 14 and day 21 from the first day of the immunization. To confirm the transfer of serum anti-myostatin antibody in serum to egg yolk, we performed ELISA with egg yolk IgG. The ELISA shows that peak anti-myostatin antibody in egg yolk around day 21 to day 28 from the first day of the immunization. After the confirmation of anti-myostatin antibody in serum as well as egg yolk, 33 eggs were collected from each group between day 24 to 34 from the first day of immunization and were incubated. And the third experiment is post-hatch growth study. All the chicks were reared up to 11 weeks of age. After 11 weeks of age, they were euthanized as per university guidelines, and all the production parameters were noted. The data were analyzed by ANOVA using GraphPad Prism software. The growth performance data shows that myo-2 group of birds were significantly smaller as compared to that of control group of birds. Similarly, male and female from myo-2 group of birds were also significantly smaller as compared to the male and female of the control group of birds. But no such difference was seen between control and recombinant myostatin immunized group of birds. Other carcass parameters like dressing percentage, carcass weight, thigh muscle weight, and breast muscle weight were also significantly smaller in myo-2 group of birds, while there was no such difference between control and recombinant myostatin group of birds. But the breast muscle weight among recombinant myostatin group of birds were significantly smaller as compared to that of the control group of bird. As there was no change in growth performance of recombinant myostatin group of birds, we performed the binding specificity of IgY from serum and egg yolk towards the bioactive myostatin through Western blot analysis. And Western blot analysis result shows that the IgY from serum and egg yolk have minimum affinity towards bioactive myostatin. So at last, we concluded that our immunization appears to fail in producing antibody having strong affinity to bioactive myostatin, resulting in no change in growth performance of the recombinant myostatin immunized birds. So our future study is to work on developing antigens capable of strong induction of antibody against the bioactive myostatin. Thank you.